I'm going to end this piece on toxicity test by looking at the botulinum toxin potency test. This slide is taken from the Canadian Council on Animal Care. You can see that the conventional test method is the mouse LD50 assay. Before we look at the Canadian Council on Animal Care's website, let's first look at a website called Nature Watch, which gives an overview of Botox. Botox is a cosmetic product used to treat wrinkles, and it is derived from the botulinum type A toxin. Botulinum type A is a bacteria responsible for botulinum poisoning. It causes paralysis, and if you get it from infected food, especially from poorly prepared meats, you will experience vomiting, stomach cramps and diarrhea. Although Nature Watch isn't an academic website, so therefore shouldn't really be included in an academic work, please grace me the privilege of quoting this non-academic site as I end this piece on toxicity tests. Saying Nature Watch isn't strictly an impartial academic organisation isn't entirely true, because they do reference Frame, who produces the excellent academic journal Alternatives to Laboratory Animals, otherwise known as ATLA for short. So this slide from Nature Watch reads, "Over the past few years the use of Botox to erase wrinkles has steadily grown in popularity. It is estimated over 100,000 treatments are done in the UK every year." And starting on the bullet points, every batch of Botox used in cosmetic procedures is tested on 100 mice using the LD50 test. This continues despite the UK and EU ban on the testing of cosmetic products on animals, and despite the LD50 test no longer being used for testing cosmetic products. Frame, whose report Growing Old Disgracefully in 2003 first uncovered the use of the LD50 test for botulinum products for cosmetic use, have now also reported a notable increase in the use of the LD50 test. And the last bullet point reads, "Estimates indicate around 50% of the botulinum toxin produced is destined for cosmetic procedures, necessitating further animal testing." And below that it reads, "The continued use of animals to test Botox destined for cosmetic purposes is in direct contradiction to the UK government's own legislation banning the use of animals to test cosmetic products or ingredients." And the last three bullet points of this slide read, under the title Further Key Facts, "Botulinum type A toxin, from which Botox is derived, is a biological product produced in batches by fermenting the same bacteria responsible for botulinum poisoning. Though no longer used to test cosmetics, the LD50 test is still used for batch testing of biological products. In order to test the potency of each new batch of toxin, the toxin is injected into the abdomens of mice to determine the amount of toxin that kills half of a given test group of mice. It's called the LD50 test, as we know. And this procedure can take up to 3 to 4 days. So now returning to the website Canadian Council on Animal Care, we can see in the left hand column the conventional test method, as has just been described, is the mouse LD50 assay for botulinum toxin potency testing. And in the second column you can see the alternative tests to the LD50 test. The first one is called endopeptidus assay, or SNAP25. The second one below that reads endopeptidus MS assay. This slide gives more alternative tests to the mouse LD50 test. The first one reads mouse hind limb assay. The second one reads mouse abdominal, I think that's a silent P, so that's ptosis assay or flaccid paralysis. And the last one on this slide reads non-lethal clinical endpoint in the mouse LD50 assay. And in this last slide, the alternative tests are mouse frenetic nerve assay, the rat intercostal neuromuscular junction assay, and the last one, in vitro cell based assay. So let's have a look at a few of these tests in a little bit more detail. We're going to look at 4 alternative tests. The first alternative test method we're going to look at is the endopeptidase assay, SNAP25. It says BONT endopeptidase assays are based on the in vivo intracellular mode of action and require as a substrate or a synthetic or recombinant peptide that contains the endopeptidase binding site. The SNAP25 assay has been validated to verify potency as a measure of consistency of clinical samples containing Botulinum type A. The assay has been set up for serotypes A, B, C, E and F. The validation status in the UK has been validated to verify potency by the National Institute for Biological Standards and Control, and that was in 1999. However, in the US, the Inter-Coordinating Committee on the Validation of Alternative Methods workshop panel concluded that it is possible in principle to use an endopeptidase assay for estimating botulinum concentration in a pharmaceutical preparation. However, they cannot currently be used to replace animals for botulinum potency testing or for detecting botulinum in environmental or biological samples. And it says future activities were recommended, and that was in 2008. Any budding scientists would have noticed that it's not just used to estimate the potency of Botox preparations. These tests are also used to make sure that medicines and test tubes, equipment, all that kind of thing, also, as with food of course, isn't infected with botulism. In this way, the test is a bit like the pyrogenicity tests, looking for endotoxins. Of course, here they're looking for whole bacteria, whereas with the pyrogenicity tests, the endotoxins are just tiny bits of bacteria, not even the DNA of bacteria, just the outer casing of the bacteria in some cases. Anyway, I thought I'd just point that out, just in case there was any confusion. And finally, under regulatory status, we can see that in the EU it is listed in the European Pharmacopoeia, and in the UK it is routinely used by the National Institute of Biological Standards and Control. In the US it has no regulatory status, and under effect or potential effect on animal use it would be a full replacement test. The second alternative test I'm going to look at is called the mouse frenetic nerve MPN assay. It reads: a frenetic nerve preparation is isolated from the mouse diaphragm and placed in Krebs Ringer solution where the resting tension of the diaphragm is measured. The incubation medium is then exchanged for toxin containing medium and the diaphragm tension is monitored until there is a 50% decrease in amplitude. This information is used to construct a dose-response curve. Under validation status it reads: in the US, a workshop panel recommended that further validation studies be concluded, and that was the ICCVAM workshop panel. In Germany it says it's been validated by the Medical School of Hanover. Under regulatory status it says that in the EU it is listed in the European Pharmacopoeia, and its effect or potential effect on animal use. It says reduction, at least 50% decrease in numbers of animals used. And under refinement it says uses killed instead of live mice. So this may sound pretty horrific, but compared to the LD50 test, this is going to heaven. The third test I'm going to look at is called the non-lethal clinical endpoint in the mouse LD50 assay protocol. It reads, this method for the mouse LD50 assay uses stage 3 clinical symptoms: severe indrawn scaphoid abdomen with respiratory distress in the form of hiccough and pupillatory dilation, in brackets it says eyes wide open bulging, in place of death as the assay endpoint. Under validation status it reads Canada, validated by Health Canada. In the US, the ICCVAM workshop panel recommended that an international validation study using the same non-lethal endpoint be conducted. Under regulatory status it says that in Canada it's accepted for use. Effect or potential effect on animal use, it says refinement. Does not rely on death as an endpoint. So here we are at the very last slide of toxicity tests. Before I read the last slide, I'll state for the record that I'm ethically against animal tests because I regard them as an abuse of power. Let's face it, the animals are forced to undergo these tests. On scientific grounds, there seems to be a case that the results gleaned from toxicity tests aren't as predictive as we are led to believe. Could it be the case that human health might actually be improved by rejecting toxicity tests using animals to toxicity tests based purely on information gathered about the specific species of interest, in our case humans? This would at least eliminate the dangerous problem, and in my opinion the insoluble problem, of species variation, i.e. species differences. Anyway, this last test is a completely non-animal test. Under name and description it says in vitro cell-based assay. It reads, the release of neurotransmitter from embryonic spinal cord neurons after 16 hours exposure to botulinum A and the reduction in inhibition of release when antibodies neutralize botulinum A are measured. Under validation status it says the ICCVAM workshop panel found that no cell-based method could currently be used to reduce or replace animals for botulinum potency testing or for detecting botulinum toxin in environmental or biological samples. In their current state of development, cell-based methods are not sufficiently sensitive. Which cell types and or cell lines are the most appropriate is also not known. Under regulatory status it doesn't have any. The effect or potential effect on animals would be replacement.